Okay, well, it's a pleasure, everyone, to welcome Dr. Alison Feder, who's coming to us from an interesting journey. So she did her undergrad at Penn and worked with
Joshua Plotkin
was there while she was doing her undergrad. And then she went and had a brief interlude in Oxford and did a master's, but ultimately ended up going to Stanford to do a PhD. And there she worked with,
Maybe you can
I'm blanking on the name.
Dmitry Petrov.
Dmitry Petrov, right? Who, if everybody's on Twitter, will know. But also, if you know his science, it's pretty cool too. And then she got a Miller Fellowship and was working with Oscar Halacek, and Monty Slatkin. And many of us know Oscar and his cool work. So, lots of interesting people that she's interacted with. And of course, we invited here primarily because of the cool work she's been doing.
Yeah.
That she's been doing in her assistant professor position at University of Washington. And she's also got a position in at the Fred Hutch. So we're eager to hear about it. And if anybody was on Twitter or on any social media that saw some of the cool animations that came out of her recent paper there, which I'm sure we'll get to at some point today. But believe it or not, she has some adaptive therapy aspect to some of her work. Aspect. So we're eager to hear it, and it's lovely to have you here.
Cool. Well, thank you for having me. I'm super excited to be here, and the reason is because just as Sandy mentioned, I don't come from a background of cancer. I know a lot less about cancer than basically anyone else in this room, certainly less than anyone else in this room. I'm an evolutionary biologist, and I'm really interested in applying things about evolutionary biology to better understand and the evolutionary history of cancers, ultimately towards some synthesis that could allow us to treat them better in the future. And the lab in general is focused on, let's see if I can maybe, it's okay, you can interpolate what you think the end of the sentence is likely to be. I'm interested, my lab works on intrahost evolution, and the reason is that intrahost evolution is, I had a wonderful discussion with Joel a few hours ago about how watching evolution happening within a person is a way where you can see evolutionary processes happening repeatedly. But another really important reason is that intrahost evolution is just a fundamental driver of disease outcomes. So if we think about the types of additional traits that populations inside of us, like cancers or pathogens, can acquire, these are things like immune escape and drug resistance, growth in new locations, motility. And these are all functionally really important features that drive whether diseases are, cleared away or whether they persist and cause long-term complications. And the particular angle that I take on this question of intrahost evolution is I'm very interested in the way that space, intrahost spatial structure, can shape the processes that happen within us. And there's sort of two major categories in which I think about this. So space both mediates biotic interactions, so whether populations like this red one are competing and have their growth inhibited by the yellow one all around it, or cooperative effects where maybe these yellow and red ones grow better together than they would separately. It also is a major form of gene exchange, less so in cancer, but in other systems where, although I think passing around elements can be an important way in which some of these traits are transferred. But space, of course, almost tautologically mediates the environment in which these populations exist. So there can be factors like local adaptation, where populations are sort of just evolving to the particular space in which they live. It can also mean just access stochastically to different resources or the environment. And then the demography of populations can be really important in terms of trying to infer evolutionary histories if you don't account for how these things have been moving. And so for a long time, and this is a discussion that I think we've touched on with a few of you earlier today, a lot of the ways that we have historically sampled populations, we lose spatial information. We take a blood draw or a nasal swab or do a single biopsy because those are the things that we have access to. But as we are increasingly able to see these processes organized in space with new technologies or sort of a better recognition that this is an important thing to sample, so more effort to sample it, we're in a new era where we are finally able to look at some of these processes resolved in space. So a big emphasis of my lab is trying to figure out what we do with the spatially resolved data and make sense of it and see how space impacts evolution and how we can sometimes use spatial information to infer evolution better than we could otherwise. So my lab works across a bunch of different systems. So we work across different levels of spatial organization, thinking about cellular processes to tissue architecture to organ systems, and also across a bunch of different systems, including viruses and bacteria and cancer. So it's a lot of things kind of going on at the same time. But what I hope to convince you of in this talk is that lessons from each of these systems are transportable. That techniques from these systems are transportable, that ideas from these systems are transportable. And through the transport of those ideas, we might be able to achieve some greater, deeper understanding than we would if we were thinking about these separately.
So, will we be bridging scales?
We're going to do some bridging of scales. There's a little bit of bridging scales at the end, but we have some other projects that are sort of more explicitly thinking about how cellular phenotypes go up to sort of spread across different tissues, which I would love to chat with you about. So the structure that I'm going to take is, if I have time, I'm going to talk about two projects. So one of them is thinking about how ideas from cancer evolution and cancer biology, really adaptive therapy type things, inform the way that we think about poliovirus, which is a little bit of a strange connection. And then I'm going to turn around and go the other direction and think about how some techniques from viral phylogenomics and macroevolution can inform the way that we analyze cancer data. So some of you were at this AACR special meeting and you kind of already heard about the second direction. So I've decided to put that at the end, just in case we get wrapped up in conversation in the first one and we don't get to it. And I would really, I'm super excited to share this first project with you. It's in progress. It's something where we would love feedback. We kind of have the rough outline of where we think the story is, but like, this is just sort of the absolute perfect audience for it. So I'm curious to hear what you think. Okay, so what can cancer evolution teach us about how to treat viruses? So I don't need to explain adaptive therapy to this audience, but one of the things that's, of course, we understand is that adaptive therapy relies on interaction between different agents. So here we're looking at a nice paper from Moxie, where we can visualize that sort of the interaction of these resistant cells competing against sensitive ones is the real thing that is keeping this population in check, and that without this interaction, the sort of whole strategic idea behind adaptive therapies, it doesn't really make a lot of sense. So exploiting this interaction, this competition, is one of the things that has shown this amazing promise. So you might think that viruses are sort of in this different camp because they don't necessarily have a lot of interaction, right? You get a virus gets into a cell, it can't replicate it on its own. And what this does is it kind of spatially separates this particular viral particle from all of its neighbors. However, we're increasingly understanding that actually this isn't the way that a lot of viruses work. That you can have viruses where you have multiple different genotypes enter exactly the same cell, and they can interact within that cell in ways that are important for their evolution. So there's all sorts of really exciting work, and sometimes you need multiple viruses to actually even enter a cell. You need sort of phage entering bacteria, need to sometimes have multiple waves of viruses before you can actually get past host defenses and get into cells. So there's a new appreciation, I think, in the field for how social viruses can be. And so this can lead to a couple of different things. So one of the things is that this can lead to gene exchange between different viruses, and this can be important in terms of shaping how much recombination plays in terms of the creation of new diversity generating processes. So, but another thing that I think is actually even the more interesting part is that it can shape the phenotype of the virus itself. So you can see here that this is a virus that has been infected by two different, a red and a blue virus, and the outside of the virus is actually composed of both of these different components. And this is the style of
European and ask what you mean by phenotype of the virus.
Yeah, so I will give you some specific examples in a second. So there's one example that I'm not going to tell you about, but it's something that we recently just had the paper accepted, and I'm very excited about, in which the phenotype that we see is actually, in this particular instance, is the ability to move between different neurons for brain infections of measles virus. And this is something where you need multiple different types of mutated viruses that have different are expressing different proteins, and the combination of proteins expressing these two different types actually work together in order to, or they appear to be working together in order to enable fusogenicity between different neighboring neurons. And there's another mode of interaction. The phenotype there is the ability to spread between cells. Here, the phenotype is the ability to be bound by a drug, and I'm going to tell you more about it in just a moment.
Cool. Would you be okay if I tweeted out a link to our YouTube channel because it would be live, or would you rather we didn't do that?
That's fine. I mean, I think if anyone else is sort of out in the world working on poliovirus and Picapavir, I'm not worried about being scooped on this project. I think that this is a little bit niche, but I think I think it's in its correct niche here. Okay, so yeah, we're going to be talking about polio. So poliovirus, despite the fact that we think about this as a disease that is, not a major threat in the United States, is actually remains a global health threat. And, it's been on the rise, especially in recent years. Vaccination efforts are waning. And you also may not know this, but we have no way to treat poliovirus. We don't have any antivirals that target poliovirus or that are FDA approved for poliovirus. And so there's sort of I wouldn't say that an urgent need in the same way that I think there are for maybe, new creative cancer approaches, but, this is certainly an unmet need, is the ability to treat poliovirus. So once you get poliovirus, it's up to your body to clear it via the immune system. And it, as I think famously, has these long-term, really devastating consequences if left unchecked or if not cleared. So even though we don't have any FDA-approved antivirals, we actually have one investigational antiviral called Picapavir. It's had a couple of different names over the years, but I'm going to call it Picapavir throughout this talk. And Picapavir is a capsid inhibitor. So normally I kind of drew viruses as sort of injecting their genetic material, but sometimes what happens is viruses get into cells and then they need to uncoat in order to be able to start their round of replication. But what Picapavir does as a capsid inhibitor is it basically binds to the outside of the viral structure, it stabilizes it, and then it can't uncoat properly, and this means that the ongoing replication doesn't happen. So Picapavir is really fascinating to me. And the reason that Picapavir is so fascinating is that it exploits the same sort of evolutionary not evolutionary principles, but it's sort of at the mercy of evolution in a way that prevents resistance evolution, or we think might prevent resistance evolution through a process called dominant drug targeting. Okay, so dominant drug targeting exploits the idea of it's sort of a specific targeting mechanism where you're trying to understand particular targets that are going to be less vulnerable for resistance evolution. So the idea is Picapavir, or a poliovirus gets into a cell, it replicates itself, and even if it gets a resistance mutation, which it does with a relatively high frequency, when these viral proteins, or when these viral genomes are making different components, for example, of the capsid of a cell, of a virus that's going to leave the cell, it's making both resistant and sensitive ones, but there's not a strong association between the genotype of resistance and the phenotype of resistance. So what happens is that all of these different capsid subunits assemble into hybrid capsid structures and then sort of randomly load the DNA or the RNA that is present in the cytoplasm. And as a result of that, even if you get a resistance mutation happening, it doesn't necessarily confer a fully resistant phenotype. You get basically suppression of this resistant type by the sensitive ones that are present. Does that make sense? So what this means is that Picapavir can come in, it can bind all the sensitive ones, and even if it can't bind the resistant ones, it doesn't matter because by the time this gets into a new cell, it can't uncoat because there's enough Picapavir that it stabilizes. So we think that this should be a really exciting way of we think that this should be a system that is not as vulnerable to resistance evolution as something that really strongly associates a genotype with a phenotype. Okay, so what's our evidence that Picapavir is actually behaving this way? Oh, I should also mention that this was about sort of a de novo resistance mutation, but if you have mixed infections of resistant and sensitive types, the same sort of principles should apply because all of the genomes are kind of competing against each other. So our evidence that Picapavir actually exhibits the behavior that we like is sort of as follows. These are some experiments that were done in Carla Kierkegaard's lab at Stanford. And what they did is they first, cultured resistant and sensitive cells, and they found that in the absence of Picapavir, the susceptible cells grow a little bit better than the resistant ones. Maybe there's a small cost of resistance, but it's relatively minor. And in the presence of Picapavir, the susceptible cells or the susceptible viruses get knocked down a lot. Okay, but this is just it shows that it's an antiviral and that it's working. What's the evidence that there's actually interaction? So they did a series of other really interesting experiments where they manipulated the multiplicity of infection of resistant and sensitive cells. So how many different resistant cells or viruses are getting into a cell and how many sensitive ones. So they started with this experiment where they took no susceptible cells and just resistant cells and said, how well do they grow? They grow pretty well. But then they said, okay, what happens if we add some susceptible cells back to the resistant ones? And we see that it actually knocks down the viral yield a little bit. So we've added more virus, but we have recovered less of it. And it turns out that as you raise the amount of virus, of the susceptible virus that you add to the system, it means the total amount of virus you get out continues to go down, and it's sharpest. The declines are most major as the MOI starts to get high. And then on the other hand, if you sort of look at this without picapavir, you see that if you add more virus, you get more virus. So this is pretty compelling. It actually suggests that the viruses are getting into the same cells, they're interacting, and then the susceptible is sort of messing up the resistant ones. Okay, so this is very exciting. Why is this not an FDA-approved drug? Well, there's been one clinical trial in humans. So this isn't a cell culture experiment, where they've just, they're passaging these things on plates. What happens when you put it in a human? So these are people who are given an oral polio vaccine, it's a live vaccine, and then treated with either a placebo or a picapavir. And on the x-axis, what we have here is the day that the individual cleared infection. They're basically monitoring them closely over time. And this is the number of participants that cleared on that day. And so you can see a couple of things that are sort of striking and not that great. So one of them is that in the placebo group, there are some people who actually did end up being recovered with resistant virus, despite the fact that there was no drug. The other thing that was sort of maybe not so great is that among the people who were given picapavir, there was sort of a bifurcation of outcomes. Many of them cleared faster than the people who got the placebo, which is awesome news. That's what you'd love to see. But among the people that did not clear fast, there is this totally opposite thing where not only do they not clear faster than the placebo types, but they also seem to get a lot of resistance evolution. This is this drug that's supposed to, by its very nature, be not evolution proof, but evolution resistant, and it seems to not very well.
It's surprising though. I mean, if you apply the drug, obviously whatever comes back needs to be resistant when you apply the treatment, whereas in the one you don't have treatment, so whatever comes back is susceptible, right?
Yeah, no, I think it's definitely, so here, so it's literally not doing anything with the placebo. Yeah. But I think what's surprising is the idea that despite the fact that we think that how is resistance emerging? Anytime a resistant mutation is happening, it's happening in the context of sensitive cells. So those things shouldn't be able to spread, but clearly they do.
Is it hard to acquire mutations that allow you to become resistant?
Very easy. It's like 1 in 10,000 mutation rate.
And they're just as fit.
Sorry?
And they're just as fit.
They seem to be, there seems to, maybe there's a small fitness cost, but they couldn't find anything statistically significant in those original cell culture experiments. So, I mean, I guess maybe there are two questions. Yeah. One of them is this surprising, and the other one is this, like, how can we explain what's going on here? And I think we'll get to both of those. And I also expect that this is an audience that is likely to be less surprised by this result than maybe others. Okay, I'm blocking Alex, but so, my graduate student, Alex Robertson, joined the lab, and he was interested in trying to understand this. He was, trying to wrap his head around why does this seem to work so well in cell culture, but it seems to select for resistance? Because the whole idea is, there should be interference, and it should lead to suppression of resistance. So what Alex decided he was going to do was build a model of poliovirus replication with picapavir to try to explain these results. Okay, so just to quickly take you through Alex's model, what he does is he starts with a viral population that has, resistant and sensitive cells in some proportions. Generally, he starts with all sensitive cells. These viruses are able to sort of insert their genomes into the cell. The genomes replicate themselves, and the genomes also have a certain probability of acquiring resistance mutations, which happens at a relatively high rate. These things create a bunch of different capsid subunits, and these capsid subunits assemble randomly. So this is basically just sort of a Poisson process, like Poisson distributed variable with the number of viruses over the number of cells. There's sort of replication is constant between these two. We don't impose a fitness cost. Mutation is just a binomial process, and then they assemble again binomially into these 60 subunit capsids. And then when they leave the cell, or they're randomly loaded with either resistant or sensitive genomes, and then they're exported outside of the cell. And so at this point, we model picapavir binding and neutralizing some of these viruses. And our data on being able to do this is not great, but I'll talk you through how we've thought about this. So we assume that at the high level, if you're entirely resistant, there's a very low, we assume that picapavir is never going to neutralize you. Other processes can neutralize you, but picapavir won't. And then on this other far end, we assume that, or sort of, if you add sensitive subunits back in, then there should be this sort of inflection where at lower numbers of subunits, you should get more and more binding, and this should lower your probability of being able to successfully encode. We model this sort of inflection point at 40 subunits, so 2/3 of the virus, of the capsid subunits are resistant. And the reason that we do this is based on measurements from this original paper where it seems like it needs about that many capsid subunits to really stabilize it. And then on this far end, we assume that if you're totally sensitive, then you're likely to be bound or neutralized. An important note here is that we don't actually bring this down to 0, and we have some sort of very low intercept. And the reason that we do this is from these experiments, we know that there is some survivability of susceptible cells even in the presence of Picapavir. So what we assume is that certain viruses are being removed from the population by Picapavir. The ones that are remaining are going to be the next sort of round of infection. And we've modeled this as sort of imagine like a transfer experiment where you take all of the viruses that are there, you plate them onto some new cells, and you watch what happens. Okay, so what Alex wanted to know first was this is, a model where we haven't really made any strong assumptions about what's going on here. It's just exactly how you would think to model this process. And we wanted to ask, does this explain the dynamics that we see in cell culture? So again, this is the sort of data that I showed you previously where we know that adding more drug or adding more virus leads to sort of this decay. And under Alex's model, it basically does exactly this. It's sort of remarkable how little messing around we had to do with this in order to just produce these results basically right off the bat. There are a few features that are a little bit different. The sort of decay is not exactly the same. We don't see an increase in the amount of virus if you don't have Picapavir, but overall we were sort of pleased with this and thought that, okay, maybe this can help us understand what's going on. So the other cool thing that we can do based on the fact that we have the simulated data that they can't straightforwardly do in the experiment is measure the frequencies of the susceptible and the resistant viruses within the things that emerge from the cell. So how many of the genomes are what? So if we focus in for a second, ignoring the non-Picapavir data because we don't think it's super interesting, just on this particular section, and focusing first on this final category, this sort of 100 versus 10. So we put in a certain amount of resistant and susceptible, and what we get out seems to be actually pretty close to those proportions. So 10 times as much susceptible as resistant, and we see sort of 10 to the 5th, 10 to the 4th, it's pretty close. So it seems like resistance hasn't really changed in frequency in this round of replication. So we wanted to try to visualize this just based on looking at sort of the average change in frequency after a round of replication, given the resistance frequencies. And what we kind of see are a few important features of this curve. The most important one is that at low frequencies, it looks like resistance doesn't really change in frequency in a round of replication, which is great. That's exactly why we think this dominant drug targeting should work. You get this kind of interesting effect at the high end where selection becomes really weak once you have a lot of competition. Once you have enough resistant viruses that the capsids are basically always resistant, and it doesn't really matter. It's hard to get rid of the selective one because it's not really costing very much. And then there's sort of this important central area where it does seem like resistance is able to be selected. And this is where there's a real difference. You can have, resistant genotypes tend to be in cells with other resistant genotypes and can actually feel their own phenotype. Does that help in terms of what phenotype I'm talking about? Cool. Okay, but this is at a quite high MOI. So what happens if we start to lower the MOI to some of the other settings? And we see that kind of maybe unsurprisingly, as the MOI goes down, this sort of suppression capability at low resistance frequencies goes up. So the average ability of a virus to change in frequency is going to be much higher at lower resistance frequencies, which suggests that maybe this doesn't work so well if the MOI is not pretty high.
What's the MOI? Is that total density of the population?
Yeah, the MOI is the average number of viruses per cell.
Right. So lower number of viruses per cell leads to less resistance.
Lower numbers of viruses per cell leads to the greater ability of a population to increase its resistance frequency. And I think maybe the way to think about this in the adaptive therapy sense or the sort of competitive sense is that basically if you're surrounded by only a few other things that you're stochastically your neighborhood could be a greater composition, you're more likely to be able to sample something where you're able to produce the correct phenotype. But let's say that you're a single resistant virus and you're surrounded by 100 other sensitive ones, you're unlikely to feel your phenotype. Okay, So given this, we thought, okay, can we understand what's going on in this clinical trial? And so it seems like Alex's model is pretty good at replicating a single round of replication. But of course, what happens in humans, or what happens in anyone who gets an infection, is you have multiple rounds, and each of those different rounds has an opportunity for the system to change. I think that this is the whole idea behind dynamical modeling, is that we can get an insight into that. Okay, so what happens in a population that's initially susceptible, like we imagined was going to happen in the people who were treated with this oral polio vaccine? So we can see that we start with a bunch of different viruses. We've sort of run this in and allowed the population to get big, and we're saying, let's allow no resistance at all to happen at the front. So after a single time step, what we see is something that looks pretty good, pretty hopeful, right? We see that the resistance frequency has stayed very, very low, and that the total amount of virus has gone down, which is good. It's going to be good for the patient. What happens next? So if you take this even a step forward, you suddenly see that the dynamics have shifted a lot. And the reason for this is that when this MOI, or when this viral yield has gone down, this has affected the dynamics of the system as a whole. So it's not like what we've done is move from having a low resistance frequency and a high MOI to a slightly higher resistance frequency and a high MOI. We've jumped up from this maroon curve onto one of these other curves, such that resistance mutations are now not surrounded by sensitive viruses to the same extent. They have fewer other things that they're sharing a cell with. So if you run this forward, you see that resistance happily takes over the population. Sensitive is happy to kind of tag along behind it as the viral population grows. Okay, not so good. But we think that this is hypothetically what was happening in these clinical trials, is that the population goes down, the interference that we base this therapy on stops working because the viruses aren't finding themselves in cells that are going to interfere with them.
But why would susceptible come back up?
Yeah, so the susceptible, I think there are two things. One of them is that there's this constant sort of mutation selection balance, such that if you have more viruses, you have the constant creation of sensitive types, just as the sensitive ones are constantly creating resistant types. And as the resistant population gets big, the creation of sensitive types, scales with that. The other thing is sort of related to this question that I mentioned earlier, which is that once you get to quite a high number of resistant viruses, there's not very strong selection against the sensitive ones, because all of the, if you have 80% of your viruses are resistant and 20% are sensitive, the sensitive ones are happy to hitch free rides along with the resistant ones, because the virus isn't making any choices when it comes to packaging its genome. It's got some capsid subunits, they assemble into capsids, and then they load whatever RNA is present. Okay, so this is sort of a lot of a very crowded vector field, but the idea behind this, I think, makes it clear what's going on, which is that if you look at the total MOI and the resistance frequency, you see that no matter where you start, you kind of get swept down to really low MOIs, and then as soon as you hit this point, you kind of turn up and you go towards this higher rate of resistance. Is that making sense? See, some people kind of trace it out, but yeah, so MOI is going down, and it's actually going down really rapidly. We've had to rescale these arrows. I'm sorry that there are so many of them. And then sort of what happens is that as soon as you hit these low MOIs, you can see that the y-axis change is actually really minimal, as we would predict. At high MOIs, the ability to increase in frequency and resistance is actually very limited. But as soon as you get to low MOI scenarios, these arrows start turning up, and you're really rapidly able to jump up in frequency.
So is that regardless of wherever you start, your calculus has been calculated by changing your initial MOI resistance frequency?
In this totally deterministic model, you are doomed. It doesn't matter where you start. You can start at 0, you can start at negative MOIs, and you won't be doomed. But if you start at a positive MOI, you will end at a resistance frequency of 1. It's actually not 1. It's somewhere over here, and this is due also to this mutation selection balance. I should also note, it goes slow. It's sort of related to this question you were asking about before. The rate at which it reaches this equilibrium is not super fast, and the reason is that selection just becomes sort of abrogated. Okay, so is pacapavir doomed? So we wanted to try to see whether this was the reasoning that behind which we could explain these clinical trial results. So what we did is we took this pacapavir-mediated neutralization and we added 2 things. So the first thing is that we went from having a fully deterministic model to having a stochastic one that has only a finite number of cells. And the reason for this is that because we think inside of a person there actually aren't an infinite number of cells, we think that there should be probably stochastic forces that are impacting the way that this works. So basically at each of these steps, instead of like figuring out, like integrating over the possible outcomes from the distribution and sort of putting them all together, we're simply drawing. So like instead of having a, looking over all possible numbers within a Poisson distribution, we're just saying, okay, let's draw some numbers from a Poisson distribution. The second thing that we add is we think about the immune system, because we think that in the context of an actual human infection, the immune system, poliovirus is a self-limiting acute infection, which means it clears naturally over the course of a couple of weeks, or a couple of days or a couple of weeks. So we basically added an immune-mediated neutralization. And the way that we do this is simply saying that after some sort of time delay, which is where the immune system is ramping up, we get this kind of exponentially mediated dampening of the number of viruses that are able to survive. Okay, and this is frequently how we think about modeling these self-limiting acute infections. So we took this model with these new two improvements, the stochastic and the immune-mediated neutralization, and we ran our own trial. We said, what would we predict should happen if you were to give 43 people a placebo and 90 people pacapavir? And what we got was basically exactly what they got. Namely, I mean, it's not quite exactly the same, but this idea that there's this sort of distribution of clearance times for people who got the placebo, and then with the pacapavir treated people fall into these two categories. One of them is that it's either you get a sensitive viral population that clears immediately, and this is presumably because the population size gets really small. And if the stochastic forces at the sort of the low point of that are, if you get extinction, then you sort of end up in this blue bar, and if you don't get extinction, you end up in this red bar.
So it's purely stochastic, or you think anything about the host that explains these two different outcomes?
Yeah, that's a good question. So we have not, there are some slight differences between the hosts here that I'm not plotting, and that's basically, in the host, we have this gamma parameter, which is just the host-mediated response, and we assume that there's some variation there, that not all hosts are clearing at exactly the same rate. We parameterize the underlying distribution for those gammas from, basically, we fit it off of the placebo-mediated data from the original trial. We haven't added anything about the pacapavir here. So it's possible that, the people who cleared early with pacapavir have stronger immune responses, but actually we think that the immune response for poliovirus is only kicking in about day 3. And so we think that probably it's not it could be immune response, it could be something else, it could be, not this, it certainly could be, but we're not sure yet. Okay, so this seemed good. So we could sort of feel like we understood something about the system, that a simple model, really, again, this is a really straightforward model, seems to really straightforwardly recapitulate these pacapavir results, and then it also seems to explain these clinical trial results without very much muss and fuss. Okay, but this is sort of bad news. It means that pacapavir is doomed. We can't use pacapavir in order to try to treat people because it's doomed to failure in the same way that if you treat cancer populations with maximum tolerated doses, they're doomed to failure. So how can we update our thinking about the system in order to try to maybe do better here? And this is completely like the sort of next step was inspired by the idea that just as in cancer, you can take this idea that maybe more drug isn't always the better thing to do, and that by tamping down the drug, we might be able to achieve different outcomes. And the reason is the drug is working too well. The drug is killing off all of the sensitive viruses, and we're relying on the sensitive viruses to control the population. What if the sensitive viruses were able to survive? What if we just gave less drug and permitted the sensitive populations to remain? It kind of, doesn't sound like that crazy an idea here, but the same idea that, okay, can we do more with less? So we don't have great ways to parameterize this sort of bidirectional dose response curve. I kind of think about this dose response curve in 2 dimensions. There's the this is at a fixed dose. This is what the dose response curve looks like, we assume. Our sort of data points here are this one and this one, and that's about what we have on this curve. It could have a different shape, we don't know. I think there's maybe some biophysical modeling we could do of the binding that we haven't done, but this is sort of how we assume it's working. But there's another dimension, right? There's coming out into this dimension, we have the number of, or we have the drug concentrations, and we might assume that at higher or lower drug concentration, more viruses that are wild type are surviving. And so we didn't have a great way to model this, so we basically have chosen a fake efficiency parameter that is titrating up or down the y-axis of this distribution. So it's a relatively simple way, but we're effectively able to say, what if the large dynamics of the curve are still the same, but we just assume that the drug works less well by leaving more sensitive viruses alive. So we tried lowering the efficacy of Picapavir. So here are just a few trials that is with the dose that I was showing you earlier, the dose that was used in the clinical trials. And now we've tried saying, what happens if you knock this down? And what we see is something quite different, which is you don't prevent resistance evolution, this still emerges, but what you do is you lower the total amount of virus in the system hugely. The immune system is still responsible for clearing the system, but what we see is that if you're trying to think about, the total number of poliovirus replications that are happening inside of a person, it's enormously lower if you just give less drug. And there are people you can't quite see on this curve. These are people who cleared immediately. So there are some good things about using a really high dose. But it also leads to some individuals to have this relapse where they get a lot of resistance. And so in thinking about this, we realized that the way that your treatment approach for Picapavir should be really dependent on your goals. And we kind of looked at 3 different goals that you could have. So you might want to have the least total viruses. Maybe these are ones that could potentially get into the central nervous system and cause this paralytic polio. That would be bad. So having fewer number of replications, keeping it around for longer but at lower levels might be fine. You could have the fastest potential clearance. Or you could have the least resistance. And you might want the least resistance, for example, because unlike cancer, you actually have to worry about giving someone else resistant poliovirus that might not have any treatment recourse in that case. So the concerns in a poliovirus system with an acute infection are a little bit different than what we think about in an adaptive therapy setting. But basically what we see is sort of similar to what I showed you, that if you treat with not very much, or you treat with really effective Picapavir, you sometimes get this really fast clearance, and it leads to, in the system as a whole, the least amount of virus. But you actually get a pretty good effect up here too, where this is enough virus, sensitive viruses kept around to keep the total population contained, but efficacious enough that it doesn't allow the population to undergo its full. And it always seems to make sense to treat earlier in this instance. In terms of thinking about potential clearance times, the way that we've set up the immune system, it doesn't care how much poliovirus is there, which is maybe a shortcoming. So so what this basically means is that if you want to clear fast, your really only route to clear fast is by either having this really early clearance or having it at the point at which the viral population has already come down a lot. So that's sort of hitting it hard. So if really what you want is to make sure that you are not walking around with poliovirus, maybe it does make sense to do this, but you might risk having more viral replications in order to achieve this. And then finally, we can see that there are certain areas where, if you don't give very much virus, or you don't give very much drug, or you give it really late, you're not going to select for resistance, which makes sense, but you won't achieve some of these good outcomes that you can see in the first 2 cases.
I don't know much about the virus, but how do you transmit it?
It's oral fecal.
So is there any consideration about, this is obviously from the individual perspective, what about population?
Yeah, that's a great question. So I think the thing that you would want to add in order to evaluate this, which we haven't done, is basically a rate of shedding as a function of what the population size is. So you could imagine that most of the shedding that you get in the context of something like this most of it is going to happen when the viral population size is large, and there's not a lot of resistance around. But it could be that I'm actually not totally sure how closely related the shedding rate and the population size should be. Intuitively, it seems like they should go together, but in the context of an oral fecal route, I'm not sure.
Yeah, also you probably don't want to shed loss of resistant ones either.
Yeah, for sure, for sure. Yeah.
Can I ask before you go on?
Please.
So day of treatment, is that like when you start the treatment?
Yeah.
So why is day 6 and day 1 for the day of clearance, they're the same?
Yeah.
Confusing.
Yeah, I think that the reason, so I'm not entirely sure. There's not a huge number of simulations, so it's possible there's some degree of stochastic effects.
Right, okay.
But I think that what's going on.
There's a kind of bump there, and like if you just go along the day there, it goes up, then it comes back down. Right?
Yeah. So I think what's going on is as follows. We assume that the immune system is taking about 3 days to kick in. And so at the point at which the immune system is entering into the picture, it's doing nothing here. If you treat on day 1, you're going to be, the clearance is largely going to be driven by just your population gets small, you have some probability of clearance. But over here, when you're hitting it really hard on day 6, the population size is going to be much smaller than it was if you were to hit it on day 3. So it's like,
Yes, there's dust everywhere.
yeah, it's all good. I don't want to draw on someone
Draw
else's.
man. Yes.
Okay, but we basically, so it's like we go, this is like viral load, this is time. So it goes up, and the natural time course of infection is basically something like this, if you don't treat it. So I think it comes down to probably where exactly those things hit are probably because the day 6 of infection is where probably like the day 1 of treatment and day 6 are probably similar population sizes, which means that the per cap of your activity in those 2 instances, this is my hypothesis, I'm not sure.
Right. So it's a population size dependent thing.
I think that's right. I think that the only thing we're explicitly modeling is that the population is going to crash out as a function of where it starts and how fast it decays. And the rate of decay should probably be pretty similar if there hasn't been any picapavir applied. So they're going to crash out at the same rate. Maybe this one is going to crash out a little bit faster because it's also got this added immune system effect that's also bringing it down.
And so the reason you don't see that as you change the drug efficacy is sort of for the reasons you explained before, like that curve shape is changing essentially.
I think that's right.
Because you don't see it as you go up there, there's no bump equivalent.
Yeah, right, yeah, so I mean, in terms of
As I increase the drug efficacy, right, so.
Right, yeah, that's a good point. I think that, so basically these levels are not really clearance mediating levels. And another factor about the clearance is that the clearance is basically, well, yeah, that's a fair point. I'm not entirely sure. I guess the way that I'm thinking about it is that you need to get your population size down small. The population sizes here are going, I think that's a fair point. I'm not sure. Okay, so I think that sort of, it seems to be true that the way that you would want to treat picapavir depends on what you're trying to maximize. But in general, I'm excited about this idea that, this is something that I'll get to that in a second. This idea that understanding how the virus is co-infecting individual cells could be really important in terms of predicting the outcomes and potentially manipulating the outcomes, which I think is a story that is very much inspired by the sorts of adaptive therapy work that people are really doing here. And this is an instance of this sort of cross-disciplinary pollination where I don't think that we would have thought to come up with it. Like, it's not clear that we would have come up with this idea unless we were reading the types of papers that you guys are writing.
I guess, a single change in dose isn't the same as multiple changes in dose, right?
Right, and this is...
Have you tried that?
No, so this is all like, I was like thinking when I was listening to Kit's talk at the AACR thing about these like very clever ways of trying to find exactly the correct ways you should be treating populations. We haven't done this. We've done something extremely straightforward, extremely obvious, which is just make the drug work a little bit less well, maybe by titrating the dose down. But I could imagine that, it's possible that there actually is a way that you can try to maximize across more of these different objectives if you're able to do this in a more informed way.
Mm-hmm.
It could be that you want to, hit a little bit lower I could imagine like you basically bringing in a smoother descent into clearance. I think some of this depends on how you think about the immune system because you could imagine another situation, which is not what we've done here, where you model the immune system as responding to the viral population itself. So the immune response gets stronger the more virus you have, which isn't present in our model. If that were the case, then it might actually make sense to keep the viral population size high at the beginning to elicit a stronger immune response to bring things down. Although you would have to ask some of the immunologists in the room whether that's how these things work. Okay. Oh, sorry, I think
Alison, if I can just jump in, this is Joel for a quickie question and observation. What I thought was super cool, besides the entire work and the fact that one can manage sort of an adaptive therapy, both sort of within and between the cells, how possible do you think it would be to model kind of the game that's going on within the cells? You already showed that there's a big effect of having, one variant, one virus in 10, 120, whatnot. Is that because that fact that they're interacting with self as well as others is kind of creating either a tragedy of the commons or a public goods game?
That's a good a good point. So one thing that I think I maybe didn't state explicitly, but is the case, is that if you have a virus that has or a cell that has 10 virions in it versus 110 virions in it, there isn't a change in the total amount of output of virus. And it does seem like from the experimental data that that isn't true, that if you add more virus, you do get a little bit more virus out. And we don't currently model this. So it's exactly the case that by adding in more viruses, we are by definition, like if you are a single virion in a cell by yourself, you're going to have all of the progeny are going to be your progeny. Whereas if you're in it with 10 other viruses, you're going to be approximately 1/10 or 1/11 of the viruses. So yeah, I totally think it could be the case that we've explicitly modeled that there's sort of interference directly just based on the fact that you're biased or you're unbiased in the way that you package genomes. But you could.
No, Alison, that makes perfect sense. And actually, it has vague similarities, to models of females laying eggs in figs, fig wasps laying their eggs in figs, where it can be one female, two females, three females, and that kind of changes the dynamics going on within the fig.
Yeah, definitely. I mean, I think that it seems totally plausible that there could be other ways that things could be interacting intracellularly in a way that we didn't model, that it could be more viruses in, more viruses out. It could be the case that there is spatial segregation on the intracell level that we haven't thought about. A lot of these viruses form separate replication complexes inside cells. And so it could be that there's sort of an assortative process wherein cells are more likely to get packaged into their own genotype or in their own capsid just based on where they are in cells. We have not attempted to model this, and the reason that we haven't is all of the choices we've made in the model is basically giving resistance the best chance, or giving picapivir the best chance to work, considering the best possible case for picapivir, which is that there's free intermixing of different viruses in a cell, that there's no cost to resistance, and that the sort of interaction between these different agents is well mixed. So there are sort of two levels that we could be thinking about adding in spatial structure. So one of them could be intracellular spatial structure, where there's this assortative process. The other one could be just sort of physical spatial, like where are viruses getting in the body? We're sort of assuming this semi-mass action approach where all the viruses come out and all of the viruses go into different cells and they don't care about where they are. So we're just in the early stages of trying to model this in space, where we have kind of two different competing hypotheses about whether space should slow or speed this process. So on the slow this process, one of the things or the evolution of resistance, we could imagine that if you're basically putting your offspring right next to each other, you're going to keep the MOI locally high. And that maybe will give resistance less of a chance of being able to get out. But the alternative could be that if you're keeping your MOI locally high, you're probably more likely to be closely related to the things that are directly around you. And so if you do get a resistance mutation, you're likely to kind of get more resistance mutations in the same place than you would in our sort of mixing system. So time will tell about how this is actually going to play out in practice. This is just sort of a little simulation where we've been looking at the color is based on the amount of resistance, and you can see that this is a situation in which the spatial structure does seem to support a system in which resistance isn't able to spread, at least in this particular stochastic iteration.
But I mean, I don't really fully understand how that spreads in the body, but I presume it's through the bloodstream and interactions with the cells. And so, people typically treat blood diseases as non-spatial.
Yeah.
But often they make the mistake of forgetting that a lot of the action's actually in the bone marrow.
Yeah.
And the marrow really does have a spatial architecture. Is that true of viruses, or is it different?
It depends on the virus. So HIV we think of as it infects T cells, it spends a lot of time in the blood, it spends a lot of time in the lymph nodes. Poliovirus affects epithelial cells. So we think it's largely, it probably is in this environment in which there probably is some 2D or 3D spatial structure. But I think the question about dispersal, how do we model dispersal, is something that I think is really hard. There aren't great data on how, so there are really cool live cell imaging experiments where you can watch poliovirus burst from a cell and see it tracked over time in cell culture experiments. You can get you can estimate a diffusion coefficient in the context of that experimental setting. What does that have to do with the body? I'm not sure. if you look in the literature for trying to see how fast poliovirus is moving, you get a lot of hits that are talking about the rate at which poliovirus moves through nerves, because they're it moves through nerves extremely fast, but this is intracellularly. It's unclear. I think that we're not going to get great data about how spatially localized this is. Our current thinking about how we might try to model this is that there's sort of two different mechanisms of action, sort of an island model approach where you may have different patches of tissues where things are locally spreading and competing against each other and may interfere with each other. But then there's probably some degree of long distance dispersal where viruses are leaving this primary site and are going elsewhere and are in this new place where if they happen to be sensitive, it's going to sort of recursively start this over again. And if they happen to be resistant, they're just going to take off.
How do you get long distance?
I think that people do this modeling for microbiome studies, but like where, you're just picked up, there's constant movement sort of from the top of your like gut system to the bottom of it, and that, hitching a ride on any of those things, whether you're able to get back out into the epithelial cells. Yeah. Good question. I think it's hard to know. I think it's possible I don't think we quite have granular enough data to try to understand this. I think if we were able to get some sort of spatial sampling, maybe we could do better. But I think that poliovirus, and apparently I'm told that poliovirus is quite easy to work with in mouse experiments. So it's possible that there are things that we could do, not us, but in collaboration with experimentalists like all of you guys do so nicely. But it remains to be seen. I think that the dispersal patterns are probably extremely important in whether we see behavior A or behavior B. I could see it both ways. Mm-hmm. And I don't think that we are going to be able to know except by saying that it does look like resistance can evolve in about, a third of cases. Mm-hmm. Yeah.
Sorry, but what we see in this slide, the previous slide, there are your resistance cells here?
So basically there's sort of a grid of cells that's a little bit too they're quite small. And what you have is an initial seeding of basically throwing a bunch of viruses onto these cells. They're able to spread into these local neighborhoods, and the reason that things fade out is because the immune system is kicking in and clearing it. The reason that everything is blue is basically because there's no resistance that is present. There are other simulations that we have where you can see that, and we should have probably thrown one of these in, that you get clear emergence and basically everything is bright red by the end. And we almost never see any sort of co-mingling of these resistant, like either it's sort of intensely resistant, not 100%, but sort of, the level at which selection stops acting. But so what we're seeing here is clearance.
Actually, I was wondering, how did you model the virus in your domain? What is the source of your virus here?
So the viruses, good question. So a virus gets infected, it goes through its replication cycle, and it spits out a bunch of different viruses that are all bursting simultaneously in discrete generations from the cell. The current localization pattern is we have just sort of simply that viruses from one cell can colonize their neighboring areas, and that this is what we're seeing. And they kind of lose steam as the immune system kicks in.
So one is the neighboring area. So you didn't consider any equation for a virus, just by the stochastic rules.
Yes, exactly. And I think that figuring out exactly how far these viruses are getting is likely as crucially important in terms of what we see. And if we get these fat-tailed dispersal kernels where rarely you go a much further distance, this is much more likely to get things off on their own in low MOI environments where we really have a problem.
Can you do in vitro experiment like that? Or inject somewhere in the middle and then see how they are spread?
So I think that people have done this. You can say basically. Yeah, in a cell, on a dish, you can basically do imaging experiments where you can capture where individual virions are going and how far and what the density is. It's almost certainly the place to start, even if you don't think they're probably, I don't know, maybe adding in diffusion and then some advection or flow through the system could be in the direction of the intestinal tract could be a way to do this, but I'm not sure. I'd be curious if people have ideas how you would go about estimating these parameters when it's hard to probe them directly.
But clearly you could do a series of sort of in silico experiments where you show the relative impact of all these changes on a fixed environment to begin with, right? Because a fixed field of cells, right? Because presumably as well, the architecture of the organ location or whatever could play a role there too, if it's folded or if it's whatever.
Absolutely,
Right.
yeah. Or, I mean, I guess I'm thinking of some of Jeff's work in terms of like whether these are highly branching structures versus
Exactly,
things
yeah.
that are tubular. I think that all of these things could be important in terms of creating local neighborhoods where viruses find themselves with neighbors or without neighbors, and how genetically related they are to those neighbors, I think are really going to be the key ingredients. Okay, cool. All right, so I'm going to close up this section, which I'm going to say is one of the lessons that I feel like I personally have learned from like reading adaptive therapy papers and thinking about adaptive therapies. I have not yet thought about this in a cancer context, but in solely just hearing talks or reading papers about it, is that interactions between agents can be exploited in order to contain resistance. And I think that this is a lesson that is not just a cancer lesson. I was talking to Sandy this morning, and he told me it's actually a pesticide resistance or insecticide resistance, which is a
pest management.
pest management system, and that this is a key factor in terms of, we should be thinking more broadly about where this can be applied, and that even things that you might think of as sort of non-interacting particles might interact more than you think, and we're increasingly learning that this is the case in viruses. And then the second main lesson being that when you're dosing, less can be more, and being tactical about the way that you're applying these sorts of approaches can lead to surprisingly good outcomes. Okay, so it's 1 p.m. I think it's foolish to start a second project, but I will kind of maybe give just a 2-minute pitch so I can show one of my animations. That this sort of the second thing we're doing on the cancer side of the lab is that we're trying to use patterns reconstructed from phylogenies and simulated tumors, and trying to take tools that actually derive from decades of amazing work in the systematics literature, trying to understand species evolution and how these things can be applied from phylogenetic trees that we're measuring in cancers. And that these phylogenies are really powerful representations of evolutionary information that I feel have been, like, at this AACR conference, every single talk was about had cancer phylogenies, and it was really cool. But I think that there's more that we can do in terms of pulling out quantitative information beyond, I mean, again, not to knock what anyone is doing with phylogenies right now, but I have seen what these things can do in the context of these speciation extinction models, and I have hope that we can probably move towards this space where we can use models to try to understand how different processes shape shapes on these trees, or like topologies and branch lengths on these trees, and that we can move towards a more an inference style method where we can learn from these things. So I'm where instead of speciation, we're thinking about birth and death. So this paper is published, so I'm just going to skip ahead maybe to the citation, but the idea here is that we can actually use some of these models in order to estimate different growth rates in different parts of a tumor, and I invite you to check out the paper if you're curious to learn more. Sorry, that's Lewinson et al., who's a really awesome MD PhD student at UW. I think that the sort of major takeaway is that even though we applied it in a sort of setting to learn about edge and center associated lineage creation rates, I think that there are a lot of additional sort of settings in which we can think about other things that can impact phenotypes that can impact rates of branching, and we can read these off of trees. Okay, so to maybe close this out, trees are this quantitatively rich source of data that can be used to quantify growth dynamics, and I think we should be thinking harder, and I would love to think with all of you about how we can do this. But the sort of larger takeaway of the talk is that I think that there are lessons that can be translated between these diverse fields, and that, the cancer people, again, I think I'm preaching to the choir here. The cancer people shouldn't only be talking to cancer people. The virus people shouldn't only be talking to virus people. We should be talking between fields, and hopefully draw a few more lines on this plot. I'm not saying that this is I'm saying that a sort of a dream world would maybe have this a fully connected graph with all of the different lines filled in. And I look forward to a future in which we get closer to this reality. Okay, with that, I want to thank you for the invitation and your attention. If you want to come find me and talk about the second part of the talk, I would be more than happy to do that. But I'm glad that we got to spend a little bit of time on poliovirus, because I think I benefited a lot from hearing your takes on this. So Alex Robertson is the grad student who has been leading this poliovirus. It's in progress. So if you have ideas, my email address is here. I'd love to hear them. I want to thank my lab. That QR code is covered, but and yeah, and basically just invite you to get in touch if any of this stuff is interesting to you. If you have ideas about how maybe we can cross a few of these disciplinary boundaries ourselves. All right. Well, thank you.
Okay, we have time for a few questions.
Yes. This is pretty stupid, but how do you get that cute logo?
The logo I drew.
Oh, cool. It's very impressive.
The logo is supposed to be sort of my PhD. I was working a lot on soft sweeps, which I think is like something that is a little bit controversial in evolutionary biology about multiple beneficial mutations spreading simultaneously. And if I tell this to cancer people, they're like, what's controversial about that? It's like, of course you get multiple convergent movements towards the same thing. So this is sort of meant to represent a soft sweep of a bunch of different, this is like time and frequency, viruses, cancer, and this was before we started working on bacteria.
Is it Moller plot?
Yeah, it's sort of intended to evoke Moller plot. Yeah.
So like going back to like the dispersion question.
Yeah.
Does this virus sort of like, does it have almost like a metastatic sort of dispersion through the body sometimes? Because you imagine if you have one virion that had the resistant genome and it just went somewhere else, it's going to be 100% resistant, just sort of automatically.
Yeah.
Is that like a real sort of issue, or do you think it's more always the body?
I don't know. So I think if you had asked me this about 6 months ago, I would have been like, yeah, it gets everywhere. All viruses are everywhere. And I've seen, like, since we started working on this, we've seen extremely compelling data that at least in certain viral settings, things can be quite localized, more localized than you would expect. Like you can see clearly, like, pockets of cells that are all infected and then surrounded by cells that are totally fine. There's this guy at University of Illinois, Chris Brook, who has been making these measurements of influenza in the lungs. It actually is another thing that reminded me of Tim's paper, where you can really see that there are sort of these distinct populations that live at the little, like kind of the ends of these lung nodules, I do not know the technical terms for these things. And then there are basically strong interferon responses that are sort of coding the pathways between them. So it does seem like this is a kind of meta-population that's semi-independent with limited migration between the different settings. How much of this applies in poliovirus? We've been looking for the data and we don't have it. I think that it might be that it needs to, like someone who is explicitly interested in this might need to go in and measure it. I would love to know, yeah.
As a follow-up to the question, do you have a good estimate of the limit of infection of this virus and how that affects the stochasticity of the model?
The limit of infection, could you tell me more about what you mean?
So if I put one virus with 100 cells, most likely I don't get an infection.
Yeah.
But like as number change of this ratio, the MOI ratio, this changes, and there's gonna be a specific MOI. And it's probably going to change depending on also the absolute number of cell versus the absolute number of virus. So like, is there a minimal amount of virus that is needed for an infection to take place in any given space? Which means like one virion that has the mutated genome, even if that one sheds somewhere else, one alone is not sufficient to create any secondary point of infection. But if there is like 3, 4, 5, and one of them is resistant, that changes. But that also plays into the stochasticity because like at that point, like you can have any combination past that number that can include like the lowest number, but the highest number as well, right? So that dampens the mutated genome more.
I think that's a fantastic question. It's something we could definitely estimate. Like we could probe this in the model, but I think we could experimentally probe this as well in terms of just like looking at the number of productive infections. We do have this discount factor where we know that a lot of the virus that exits a cell is not actually capable. It can't form plaques. And so we know that there's a lot of things that are being pumped out that are virions that are probably bound and neutralized, but they couldn't lead to productive infections. In terms of and they are relatively rare. So what we're modeling is basically only ones that we think are productive, but it doesn't quite answer your question of saying like how many productive ones are needed to infect a cell. But it is accounting for the fact that we know that there is a lot of unproductive virus that can't. So I don't know the answer, but I do know that this is a phenomenon that we see in other viral settings, and so it totally could be at play here. Yeah.
Sorry, about your stochastic model, how many cells type did you actually consider for your stochastic model?
Yeah, so the density, that's another good question. Not a huge number of cells, and it's related to this idea that we don't think that this is a systemic virus that's in every cell in the body. We've been using the density of cells that were in the dishes in the in vivo setting or the in vitro setting, the cell culture experiments we saw at the beginning. But I think it's probably more viruses than that. It's probably not 1,000 times as many viruses as that. And I think basically what we still need to do is see that these results are robust across different population sizes, because I don't think we have a great idea of what the number is.
I was wondering if you actually simulate your model with some distance of virus and calculate the percentage of the resistance that happens according to this distance that you do consider for your virus. And again, do the same simulation with the same soft but different distance of the virus, and calculate again the percentage of the resistant or sensitive cells, something like that, after the end of the simulation. And that map would be a nice map to see how this distance of the virus can affect on the percentage of resistant cells that you can see on your in vitro experiment.
Yeah, that's and the in vitro experiment. Yeah, I see what you're saying. Yeah, that's a great suggestion. Thank you.
I was going to ask a question about, just thinking about adaptive therapy in cancer. One of the key questions is, what disease setting we should go for cure versus control.
Yeah.
Is that at play here, or are you thinking by lowering the dose, you still have the immune system coming in later, so you don't have that critical question, or what?
Yeah, I think it's a good question. So if you look at let me see if I can bring up that plot again, the sort of different outcomes we could be going for, it definitely seems like there are some good things about hitting hard. Mm-hmm. And one model that we didn't show here is what happens if it works perfectly. And if it works perfectly, the system immediately collapses. And so, in this sense, there isn't really, we think that pre-existing resistance mutations, they could be there, but they're likely shielded. And if it really is the case that the slope of the behavior of this when the resistance is rare is sufficiently favorable, there's no reason not to hit as hard as you can if you can actually achieve that. I think the problem is that you're never actually going to be able to hit as hard as you want. Yeah. So there are nice things. There are good properties about being down in this corner. I think the bad things is that if you don't clear, you certainly get resistance 100% of the time. And it's sort of about the trade-offs that you want to make in those settings.
Yeah.
Yeah.
Thanks for a nice talk. I am kind of wondering about the effect of aging. So I don't know anything about polioviruses. I don't know anything about viruses at all. But I'm just wondering, is it like a disease, or the polio itself? Is it a disease more common in the newborns or in children, or can it hit every single person equally well throughout the lifespan? The reason why I'm asking is, at least in epithelial cancer, it happened more often in the older people, right? And at that point, I would be expecting that the immune response itself is going to be weaker. Mm-hmm. And that going back to your model, I would expect that the effect of the immune response in order to induce clearance and so on, it would be much more delayed. If the polio itself can be occurring to patients all throughout the whole lifespan, then there must be some sort of a distribution of how immune response is attacking, right?
Yeah, it's a good question about the age structure. I don't know the answer. I was under the impression that it is primarily it tends to be younger people, and I think this is right, but do not quote me on it. I think it is like the vaccination is basically you get it once and you won't get it again. So I think that there aren't sort of factors of original antigenic sin that complicate the question. In terms of how I think I agree with your assessment that if you have a weaker immune system, and this is again something that we can test, you can try to understand whether it changes basically the outcomes here, and I imagine that it would. If nothing else, it's going to slow down the clearance times and give more opportunities for resistance to evolve when treated. But so I guess the short answer is I'm not sure, but I think it's something we could probe with a model.
Cool, thank you.
Could you clarify what is day 0 of treatment and like how that relates to the actual onset of the diseases diagnosed? Because that's gonna also affect when actually the immune system is kicking in compared to the treatment.
Yeah, that's another great point. So we were primarily interested in trying to understand the dynamics of this oral polio vaccine trial, where we know exactly when the person was infected with the virus. And in the trial, they either treated people at 24 hours or 72 hours. So in that sense, we know exactly, we can control when after treatment. In reality, you're right that symptom onset takes several days. And it actually turns out that the time course of this non-OPV infections are longer than what we see here. It takes some time to incubate, symptoms express. It's generally a short period of time of symptoms, like 3 to 5 days, but then the shedding period can be longer than that. And it's unlikely that someone is going to seek a vaccine or seek a treatment if they're unaware that they're sick. So I think that realistically, we might not be able to access some of the things that are right here. But in some ways, this could be pushing us more towards saying that these outcomes are better than these outcomes if it's going to take at least a few days before we're able to. It's going to depend on basically how entrenched the viral population is at the time of treatment. And the bigger it is, the more likely that actually treating with less is going to be the better strategy, at least within the context of what we've seen so far.
Can I ask a little bit about the drug dynamics? So,
Yeah.
A vaccine versus like a regular drug, they behave differently, right? So when you're talking about drug efficacy here, is it the vaccine or is it, it's a different drug?
Yeah, so it's an antiviral. The setup of this trial is that there's sort of two different polio vaccines. There's a live attenuated vaccine and which doesn't. I think we don't have fears of it reverting, but if you take the oral polio vaccine, you can actually it continues to shed. You can pass it into the water supply. Other people can pick it up. So one of the things we might be interested in, kind of going back to David's question about transmission, is figuring out whether we could use pacapavir in a setting where people who are administered the oral polio vaccine, which is much easier to transport, whether using it in conjunction with pacapavir could be a way to keep it from getting released into the community, to have opportunities of the opposite of attenuation, getting strong again.
And so the reason why I was asking that was more about the concept of the timing and dynamics of the drug. So traditionally, in most adaptive therapy models, we treat the drug as an on-off, or like you give the dose, it's instant, and it doesn't take any time to wash out. It doesn't linger. It's just like that, right? And so, with this, is there a time scale associated with that drug? Does it delay before? So you don't give it anymore, but it's still effective for a few days. Or?
Yeah, it's a good question, and I don't. It's possible that we know I think we do know the half-life, but I'm unfortunately not 100% sure what it is. We're basically kind of treating it as an on-off switch right now, where it's something that we can make appear.
But it could be important if your dynamics, it seems, are in a kind of quite a short window.
Yeah.
It feels like it's only a few days.
Yeah.
And so that could matter then for the half-life of the drug. But that could also potentially give you a lever to tweak in terms of giving it versus not giving it, and doing that repeatedly, right? So for getting sort of more interesting dynamic responses there.
I think that's definitely correct. And in terms of, like, we see that even, let me see if I can pull back to this slide. Yeah, here we still have quite a lot of virus. Like, the MOI has dropped pretty substantially, but I agree that, like, I think the dynamics happen pretty quickly, in part because it's this discrete generations where, you get these big changes. In reality, I think that we might also see something different if we were modeling this in continuous time, where the bursting time is going to affect who's getting in to different cells with who else. But yeah, I think that sort of by virtue of the fact that we've done it in this way, our sort of levers, we are assuming that the only thing we've played around with so far is basically you turn the treatment on and then you wait and you can turn the treatment off. We haven't tried titrating doses. We haven't But these are all things, like you can imagine turning it on and then watching the dose decline and then seeing how it would work. Because it might be that, what you want to do is just give someone one dose of Bicapavir once, and this is actually pretty good in terms of knocking out the time of the viral population when it's at the highest amount. And it could be cost effective.
I mean, it's something that we've seen in different cancers. So, for example, and really largely different drugs. In ovarian cancer, given PARP inhibitors, what we've seen there is that if you give a treatment holiday, it's actually bad, versus if you reduce the dose. And part of the reason why it's bad is that completely removing the PARP inhibitor allows the population to repair the DNA damage and just take straight back off again. Whereas if you just hold it on at a lower dose, it stops that repair from kicking in, right? So different drug altogether, but it's just that dose modulation there plays a kind of important role, as opposed to where we normally would just stop the treatment altogether, right? So it's a different type of competition in some sense. And I feel that you've got good confounders here, because the resistant cells can create susceptible cells, and the susceptible cells can create resistant cells. So you've got all of this happening simultaneously in a stochastic manner, right? So it's an interesting system to try this out in.
For sure. I think that another thing that we haven't really thought about is sort of like, once you are bound by Bicapavir, we assume that you're bound, but in reality, there's probably some association, disassociation of the drug. And so it could be that you're effectively like, you have viruses that are bound that are not doing anything currently, but if, you reduce the density of the drug in the system over time because you're not treating anymore, you're going to get more disassociation, and you could have reactivation of things that can't infect at that particular time. It's also not something we've considered. But yeah, I think that's a good point.
Any last questions? Yeah.
Well, thank you guys for indulging me in talking about a virus. I hope this has been interesting. I just wanted to say again, we've been inspired by a lot of the work going on here, and so I wanted to try to share something about how it's informing something totally different. So thank you.
Thank you.